Chapter 5 deals with the working cell. It's sort of a catch-all chapter. There's several topics that need to be discussed about the cell and cell function, and so they're put into this sort of this chapter. And so the first part of the chapter deals with energy. And energy is the capacity to do work. The cell can perform its function, its job, based on how much energy it has to its use. It moves matter in a direction that it normally wouldn't move if left alone, and it makes change possible. And for organisms to stay alive, they must have this change to occur. There are two types of energy. There's kinetic energy. That's energy actually doing work. You can think of a person pedaling a bicycle, right? And then there's heat energy, is a type of kinetic energy. Heat is the energy associated with the movement of molecules within a body of matter. Light is also another type of energy. Back to heat. All matter, doesn't matter which matter you're talking about, the molecules move. All right, so heat is just the energy associated with their movement within a body of matter. Potential energy is stored energy. It's the capacity to perform work that a matter possesses as a result of its location and arrangement of molecules. A good example of this, a lot of people, when we think of stored energy, it's energy that is not actively doing work. And if you think of water behind a dam, the water has a potential to flow if the dam wasn't there. But when the dam is breached, then the water flows, right? So a type of potential energy is chemical energy, and that's the energy that's associated with the arrangement of atoms in a particular molecule. And this is the potential energy of living organisms. It's chemical energy. So most of the energy available to cells rests in the covalent bonds of the molecules, right? So the energy that's associated with macromolecules that we've talked about are in the covalent bonds. Energy is actually converted from one form to the next. And there are two laws that govern this energy conversion. They're the first and second law of thermodynamics. The first says that the total amount of energy in the universe is constant and finite. So energy is not created or destroyed, it's just converted from one form to the next. And if you have this energy conversion from one form to another, some energy is lost into the environment as heat. So heat, we talked about what heat is. It's the kinetic energy that is the movement, is the energy associated with the movement of molecules. Second law of thermodynamics says that no energy conversion is not 100% efficient, and that energy in the universe is flowing from higher energy forms to low energy forms. If we look at the food chain again that we did in chapter 1, there's more energy in the sun than there is in the producers that are on the earth, and then the producers have more energy than herbivores, and herbivores have more energy than carnivores, and so forth and so on. So it's flowing from forms of higher content to lower content. And then lastly, the second law of thermodynamics states that a measure of degree of a system's disorder, sometimes it's called the chaos, is entropy, increases over time. This entropy is a kind of a hard word. You probably heard it if you've watched Big Bang Theory on television. But entropy, what it's stating is that in the universe, that as you move from high energy potential energy, as it flows through the system, then what happens is you get an increase in energy that is not really usable. And we're talking heat at this point. So if we look at the first law of thermodynamics, we see that heat's lost to the environment, and according to the second law of thermodynamics, then entropy increases over time. And so the amount of heat energy is increasing as during the life of a system. And so we would call that entropy disorder. Life's chemical reactions store and release energy. Now, I just said that the amount of energy in the universe is finite and constant. It can't be created or destroyed, right? But there are two reactions that are used by living cells that allow them to obtain energy and then release energy, all right? And these are endergonic and exergonic reactions. So endergonic reaction is one that ends with a net gain of energy. And so the products have more energy than the reactants. If we look at photosynthesis, we start with carbon dioxide and water, and we end up with glucose and oxygen. Glucose is a lot more energy than water and carbon dioxide. So that's an example of an endergonic reaction. Exergonic reactions are reactions that end with a net loss of energy. So the reactants have more energy than the products. So if you look at cellular respiration, when we do in chapter 6, it talks about using glucose, breaking it down to release energy for cellular work. And so the glucose and oxygen are the reactants. The products are carbon dioxide and water. But getting back to the laws of thermodynamics, these two types of reactions are coupled. And so there is really still no net gain or loss of energy in the system. And this is called cellular metabolism. And so what the cell does is use endergonic reactions to get potential energy for the cell, and then store it, and then use exergonic reactions to release that energy so that it can be used for cellular work. So cellular metabolism is what is working there, right? And so you've got complex molecules in the cell. They have more energy. An exergonic reaction breaks those down, and then you have simple molecules. An endergonic reaction takes those simple molecules and puts them together to make more complex molecules in endergonic reactions. The next topic that we need to talk about is ATP. ATP is adenosine triphosphate. It's a nucleotide, and it's the key to the cellular engine, all right? And so yes, we have to have gasoline, which is glucose, to do work. But in order for the car to go, or a cell to work, in this case, we have to have the key, and the key is ATP. All right, and so the TP part is the most important here, because in ATP you have 3 phosphate groups. Right, the energy that is stored between the second and third phosphate group is where the energy that when glucose is broken down, it provides this energy then is stored in between the second and third phosphate group of ATP. All right, and then once ATP is taken to wherever it needs to do wherever in the cell to do work, that the second and third phosphate group, that energy that's between those two, is broken. That bond is broken, and energy is released to do the cell work. So the triphosphate is the important thing here. And so if we have ATP, well, let's go with you have glucose being broken down in an exergonic reaction, and that energy, that phosphate group, free phosphate group, and ADP, which is adenosine diphosphate, so it has 2 phosphate groups attached to it. The energy that is released as the glucose is being broken down, the ADP gets a phosphate group attached to it to form ATP. Once there is a reaction that needs to be to take place, ATP is broken, and it fuels an endergonic reaction that is doing the work of the cell. Okay, so the way the cell replenishes its ATP, because again the cell is a major recycler, is called the ATP-ADP cycle. And so energy input drives the attachment of the unbound phosphate group to ADP, creating ATP. When energy is needed, the phosphate group is cleaved or cut from ATP, resulting in the release of energy, and then you get ADP and an unbound phosphate group again. So this just continues, the cell continues to use ADP and a phosphate group to form ATP, and when ATP is needed for work, it is broken into ADP and a phosphate group. Another thing that we're going to talk about in chapter 5 is the topic of enzymes and how they work. Enzymes speed up the cell's chemical reactions by lowering the activation energy. All right, and we'll talk about that in a second. So enzymes are these protein molecules that we talked about in chapter 3, and they serve as biological catalysts. They increase the rate of a reaction without actually being changed into another molecule in the process. All right, and so there are 4 features that all enzymes share. 1, they don't make things happen that wouldn't happen on their own. They just make them occur millions of times faster. If the cell was, if the reaction was left up to its own within the cell, it would not work fast enough, and the cell would die. So enzymes are really important in that respect. They are not permanently altered. They're not used up. Right? They're used over and over again. Again, the cell recycles most of its molecules. The same enzyme usually works for both forward and reverse directions in a reversible reaction. So sometimes you have products and reactants you have reactants that form a product, and sometimes that product then turns around and becomes the reactant, and it changes back into the product then of that reaction would be the reactants that you started with for the first reaction. So they can go in a reversible order. And then specifically, each enzyme is selective or only recognizes a certain substrate. A substrate is a molecule that is helped during the reaction, and enzymes only recognize the molecule that they are supposed to help with the reaction. And that's called the solute. Substrate. So, activation energy is the minimum amount of collision energy. Remember that molecules are moving. That is required to bring a reactant molecules to an activated condition. We call that the transition state. An activated condition or transition state is where the reaction occurs spontaneously, all right? And so, again, the reaction would occur without the enzymes, but it's just going at a lot faster rate. And they do this by lowering this activation energy. Not raising, lowering the activation energy of a substrate. So there were these things called Mexican jumping beans you could buy in like the back of magazines. And if we put some Mexican jumping beans in a beaker and just sat there and watched them, they would start bouncing around each other, and at some point one would jump over the edge of the beaker and into a bowl that you sit next to the beaker, right? In this example, the beaker side would be the activation energy. It's the amount of energy that is required for the Mexican jumping bean to jump into the bowl, right? And so if you let it go, those Mexican jumping beans would finally all jump into your bowl, right? But if we were to lower the activation energy, therefore in our example lower the edge of the beaker, then those jumping beans would jump into the bowl a whole lot faster than they would if you just had the beaker wall at its given level, all right, and at the original level. So that's what enzymes do, is they lower the barriers, the energy barrier that is required for the reaction to occur. And so enzymes have a substance or a molecule that it acts upon, and that's the substrate. And each enzyme has a crevice or a hole or an area in the surface of the protein that is the place where the substrate is recognized and fits into. And once that substrate fits into the active site, then the activation energy can be lowered. And so if you think of Pac-Man, right? And Pac-Man's mouth is open, Pac-Man would be the substrate. Would be the enzyme. The little triangle wedge that it would fit into his mouth would be the substrate. That's the molecule that that the enzyme recognizes. When that triangular pie-shaped part fits into his mouth, then that mouth area would be the active site, right? and once it fits into the active site, it is fully satisfied. It ate its little food, right? So this leads us into the induced fit model. Is the active site recognizes the substrate, but the substrate doesn't fit exactly into the active site just perfectly, and so it has to kind of work its way in, and as it does, hydrogen bonds are broken and reformed, and once it fits in there exactly right, then the transition state is met, and the reaction can occur, right? And so so the induced fit model sort of gives you a little bit more depth idea of how that activation energy is lowered and the transition state is met.